Good morning, students. I am Professor Nitesh Sharma. Students, today I am teaching you extra embryonic membranes development and functions. Students, this topic, extra embryonic membranes development and functions, is included in the paper 2nd Developmental Biology of BSc Part 1st. Students, these extra embryonic membranes are very important because their role is very important in the development of embryo. Students, an extra embryonic membrane is one of the membranes which assist in the development of the embryo. Such membranes occur in a range of animals from humans to insects. They originate from the embryo but are not considered part of it. They typically perform roles in nutrition, gas exchange, and waste removal. Students, there are four standard extra embryonic membranes in birds, reptiles, and mammals. The yolk sac, which surrounds the yolk, the amnion, which surrounds the cushions, the embryo, the allantois, which among avians stores embryonic waste and assists with the exchange of carbon dioxide with oxygen, as well as the resorption of calcium from the shell, and the chorion which surrounds all of these and in avians successively merges with the allantois in the larger stages of egg development to form a combined respiratory and excretory organ called the chorioallantois. Students, the four extra embryonic membranes are yolk sac, amnion, allantois, and chorion. These membranes are formed outside the embryo from the trophoblast only in amniotes, that is, in reptiles, birds, and mammals, and perform specific functions. Students, now you can see in this picture how these membranes develops and do their necessary functions. Students, now we are going to discuss the development and functions of extra embryonic membranes. First, yolk sac. It is formed of splanchnoplure, inner endoderm and outer mesoderm, and is well developed in reptiles, birds, and protherians having polylacethal egg. It is mainly digestive in function, so acts as extra embryonic gut. It also absorbs the dissolved yolk and passes it to developing embryo. In human beings, it is vestigial. Students, now second one is amnion. It is innermost fold of somatoplure, inner ectoderm and outer mesoderm. Above the embryo, between the amnion and embryo, there is amniotic cavity filled with amniotic fluid secreted by both embryo and amnion. Amnion protects the embryo while amniotic fluid acts as shock absorber and also prevents desiccation of embryo. So, students, now third one is allantois. It is a fold of splanchnoplure developed from the hind gut of the embryo. It is well developed in amniotes and polylacethal eggs, example reptiles, birds, and prototherians, and stores the nitrogenous wastes of the embryo. So acts as extra embryonic kidney. In most of eutherian, it combines with chorion to form allantochorion, which takes part in placenta formation. Allantoic placenta. It is reduced in human beings. So, students, now the last one is chorion. It is the outermost fold of somatoplure and surrounds the embryo in reptiles, birds, and prototherians. Allantochorion acts as extra embryonic lung and helps in exchange of gases. But in primates, including human beings, only chorion forms the placenta, that is chorionic placenta, while in other eutherian, allantochorion forms allantoic placenta. So, students, you have listened the lecture about extraembryonic membranes, their development, and how they functions. So, thank you very much for your patience.